Excellent. Thank you very much. Thank you very much for the opportunity to present our work on ALS. I'm a researcher at Karolinska Institute studying how blood vessels and their dysfunction can contribute to ALS disease onset and progression. Right. So ALS is known for its dysfunction of the nerve cells that are connected to the muscles. So the moment they start dying and disconnecting from muscles, we start to see the onset of symptoms in patients. But there are several things that we still don't understand about ALS, and particularly why some people do develop ALS and some don't. What would be the mechanisms behind the very early phases of ALS before you see the doctor? What's happening before the first neuron is dying? And also a big problem in treating ALS is that we really cannot predict very well how long an individual patient will live, because the life expectancy can vary from several months to several years, and it's very, very difficult to predict that at the onset of the disease. So if we think of a brain as our big computers that are hungry for energy, computers run on electricity, and brains run on energy that is brought by blood vessels, because it is the blood vessel that is delivering oxygen and sugars and glucose for the brain to use to function. So we think that the moment there is a problem at the blood vessel level, perhaps this is the kind of a problem that would make ALS worse or happen earlier. When we're thinking about the proper functioning of a blood vessel, is that there should be a well-regulated dynamics of a blood flow in the brain. These blood vessels should be stable. They shouldn't become broken. And we see that these events of decreased blood flow, decreased glucose uptake, and vessel remodeling are happening in ALS patients. So in order to try to understand that, we looked at the mouse models of ALS. Eva and Peter were already describing that you can put a mutation that creates the inherited form of an ALS into a mouse, and then that mouse will develop ALS. So then you can look at this disease in any time point you want. And the brain is built of several building blocks, several cell types. For example, we have the neurons that are connecting, we have microglia, we have astrocytes, and then there are the cell types in blood vessels shown on top. So there are endothelial cells and fibroblasts and other cell types. So we were trying to interpret the activity of those cells in the brains in mouse models and in human ALS. And the typical thing that we expect to see at the end stage of ALS is that the cells that are responsible for neuroinflammation, like microglia and astrocytes, they are overactivated. And the neurons that are dying, here shown in the dark blue, are the ones that reduce their production of genes and proteins. So when we were interpreting the vascular cells, we saw that cells called brain fibroblasts that sit on top of vessels are also activated in ALS, but we didn't know which of these events happens first and which is second. What is the beginning and what is the consequence? So fortunately, we can look at the mouse model and try to interpret which cells become active in the earliest asymptomatic stages. And we found that these vascular fibroblasts are activated long before you see first symptoms in the mouse, and they become active before the microglia, before the neuroinflammation, before we have onset of symptoms in these mice, before we have neuromuscular junction decoupling. We already see problems happening at the blood vessel. So since we knew which genes or which building blocks are activated in those cells, we tried to look for them in ALS patients. So here in red, we see two types of proteins that are accumulating around blood vessels. They're shown in red. They're called collagen 6 and STP1. And by accumulating around the blood vessel, they're disrupting its function and make it less active and less efficient at delivering oxygen and glucose to the brain. So we came with this conclusion that these cells, called the fibroblasts, are changing the structure of the blood vessel. They're shown here in red. They're secreting some proteins into the blood vessel. They're called STP1 and collagen 6. And at that point, we really didn't know to what extent does this really have an effect on ALS patients. So since these are proteins produced by injured blood vessels, we tried to look for those proteins in the bloodstream. And thanks to a collaboration with numerous clinics in Sweden and around the world, we were able to collect a substantial plasma sample biobank from ALS patients. So when we look for the vascular injury protein, called STP1, here we see an increase of the protein in red and the lower concentrations in green. And high levels of this vascular injury biomarker were associated with shorter survival in ALS patients. This makes us think that vascular injury is an independent survival predictor than other forms of survival prediction. So I think that what is important to take with this is that today ALS is likely understood based on late disease events, like neuromuscular junction decoupling and then dying of the neurons. But we don't know what's happening very early. So our study points that these vascular mechanisms could help us understand early disease events in ALS. And since ALS is highly unpredictable, by measuring those vascular injury proteins, this could become an independent indicator of ALS patient survival. And once we can apply these measurements to a larger cohort of patients and better predict patient survival at disease onset, this will help us evaluate any kind of a drug treatment. The better we can predict the survival of a patient before any treatment, the better we can evaluate the effects of clinical trials. So to summarize it really briefly, this was rather unusual for us to see that there are signs of vascular injury in ALS patients in the brain. This is shown in the left panel. When we see the individual blood vessels, we see that there is a cell type called the perivascular fibroblast that is remodeling the structure of the blood vessel. And when we try to follow the proteins produced by these fibroblasts in the bloodstream, the high concentration of the STP1 indicate poor prognosis in ALS patients, and low STP1 proteins indicate good prognosis. I'd like to thank everyone that contributed to the study, several groups in Sweden and abroad, and also the founders, Ulla-Carin Lindquists Stiftelse and the Neuroförbundet, for supporting our work. This study was published in the spring of 2021. I would be happy to tell you more about it, so feel free to contact me via Twitter or email.